Assalamualaikum warahmatullahi wabarakatuh. Hello everyone. We are from Group 4 in RKI Nurse class. Today we will present our presentation in biochemistry course with enzyme and coenzyme for the topic. Before we start the presentation, I would like to introduce the group members. There are Amalia, Khairun, Nissa, Flora, Balqis, Ervina, Leyla, Octaviani, Galuh, Cantya, Kusumawicitra, and Nuning, Retno, Anggraini. And this is the theory that we're gonna discuss in this section. First is enzyme and coenzyme with the submatery and biological oxidation, and the last is the relationship with health.
Hello everyone, my name is Amalia, Khairun, and I will explain about definition of enzymes and classification names of enzymes. The first is definition of enzymes. An enzyme is a substance that acts as a catalyst in living organism, regulating the rate at which chemical reaction proceed without itself being altered in the process. Protein that speed up the rate of a chemical reaction in a living organism. An enzyme acts as a catalyst for a specific chemical reaction, converting a specific set of reactants, called substrate, into specific products. Without enzymes, life as we know it would not exist. The next is classification names of enzymes. An enzyme will interact with only one type of substance or group of substances, called the substrate, to catalyze a certain kind of reaction. Because of this specificity, enzymes often have been named by adding the suffix "s" to the substrate name, as in urease, which catalyzes the breakdown of urea. Not all enzymes have been named in this manner. However, and to ease the confusion surrounding enzyme nomenclature, a classification system has been developed based on the type of reaction the enzyme catalyzes. There are 6 principal categories and their reaction. The first is oxireductases, which are involved in electron transfers. The next is transferases, which transfer a chemical group from one substance to another. The third is hydrolysis, which cleave the substrate by uptake a water molecule, or hydrolysis. The next is lysis, which form double bonds by adding or removing chemical groups. The fifth is isomerases, which transfer a group within a molecule to inform an isomer. And the last is ligases or synthetases, which couple the formation of various chemical bonds to the breakdown of a pyrophosphate bond in adenosine triphosphate or a similar nucleotide.
Third is enzyme structure. Enzymes are a linear chain of amino acids, which gave rise a three-dimensional structure. Enzymes are composed of 3 main components. First, main protein component. Part of enzyme which is a protein called apoenzyme. Apoenzyme or other terms is apoprotein. The second is prostatic group. This enzyme component is not protein. This is coenzyme and cofactor. Coenzyme and cofactor is very tightly arranged, even bound by covalent bond and enzyme. Coenzyme often refers as second substrate. Coenzyme has low molecular weight. A coenzyme is stable to heating. Coenzyme bound to the enzyme non-covalently. Coenzyme functions are to transport small molecules or ion, especially hydrogen, from one enzyme to another, for example NAD. And then cofactor. Cofactor is a function to change structure of the active regions or needed by substrate to bind the active region. The cofactor examples are which can be small molecules such as ferron, chrome, zinc, magnesium, mangan, potassium, nitrogen, MO, and SA. The third is enzyme active site. The site is part of the enzyme that binds the substrate. This area is very specific because a suitable substrate can adhere to or bind to the site. Enzymes are proteins that have a globular structure. The curved enzyme structure causes the presence of an area known as the active area. Fourth, character of enzyme. In general, enzymes are protein. Enzymes are almost entirely protein, and almost half the amount of protein present in cell is named enzyme. Enzymes are biocatalysts, namely speeding up a reaction but without changing because the reaction itself. Enzymes are only synthesized by and in cell. Enzymes will be synthesized if cells have the gen for this enzyme. Enzymes are compounds that are much smaller so that reaction can take place in the cell. In progress of catalysis of a reaction, the enzyme react first the substrate to form a new compound or substrate enzyme complex and will produce a product. Enzyme plus substrate is gonna be product. Enzyme can increase the reaction rate about 107 until 1013 times faster than non-enzyme catalysts. Increase reaction speed. Enzyme can reduce the activation energy of a reaction. For activation, enzymes require less energy. Enzymes have specifically properties that cause enzyme to choose the substrate to be recognized, bound, and processed further. Its activity is lost when the enzyme is denatured. For this activity, there are a need for a coenzyme and cofactor, active forms, and vitamin or minerals. Vitamin C or ascorbic acid must be in the diet because the body doesn't have the enzyme that help the formation of vitamin C.
5. How enzymes work. Two models have been proposed to explain how enzymes work. They bind to substrates. In the enzyme, there is an active site, the active site, where it attaches to the substrate during the reaction, while the inactive site is called the allosteric. E. Model key and lock or lock and podlock. This model was published by Emil Fischer in 1894, which stated that the shape of the substrate molecule with the enzyme active site was similar to the key with the key. According to the theory of lock and key, the active site of the enzyme is specific and inflexible, so the form of the enzyme with the substrate form has a specific shape to produce a product from the substrate, but the enzyme or enzyme form does not react. B. Induced fit model or FIT, a model proposed in 1958 by Daniel E. Koslan, Jr. In this model, the substrate causes conformational change on the active site of the enzyme. According to the theory of induced fit, the active site of the enzyme is flexible, although the shape of the enzyme and the shape of the substrate do not fit. The enzyme will adapt its shape to the shape of the substrate so that the reaction occurs to produce a desired product. When working, enzymes are influenced by several factors. First, concentration of enzyme. Second, substrate concentration. Substrate is a substance that is converted into something new. Generally, there is a proportional relationship between the substrate and the final product if the enzyme concentration is constant, the pH is constant, and the temperature is constant. Too much substrate and low enzyme concentration will cause the reaction is not optimal. Third, effect of temperature. Generally, enzymes work at the optimum temperature. When the temperature drops, the activity stops, but the enzymes are not damaged. Conversely, at high temperatures, the activity decreases and the enzymes are damaged. 4. Effect of pH. Change in pH can reserve enzyme activity, that is, change the end product back into a substrate. Enzymes can work optimally at a certain pH. For example, the lipase enzyme optimal pH of 5.7 until 7.5. 5. Effect of inhibitor. Inhibitors are chemicals that inhibit enzyme activity. For example, salt of heavy metals such as mercury. 6. Enzyme metabolism. Definition. Metabolism is the process of a series chemical reaction that occur in living cells of organism. Chemical reactions are divided into two metabolism. First, catabolism is a chemical reaction in living cell that aims to produce energy by breaking down or decomposing a compound. For example, is cellular respiration. Second, anabolism is a chemical reaction in living cell that aims to synthesize or form compounds or molecules by using energy. For example, photosynthetic reaction or assimilation and polymerization. The difference between anabolism and catabolism. Anabolism form or make compounds. Catabolism break down compounds. Anabolism requires energy. Catabolism generate energy. Anabolism converting inorganic compounds to organic. Catabolism converting organic compound into inorganic compounds. Anabolism, the nature of the endergonic reaction. Catabolism, the nature of reaction is exergonic.
Biological oxidation. First, definition of biological oxidation. Oxidation is the removal of electrons and reduction in the gain of electron. An example is the oxidation of various ions to various. The oxidation will always be accompanied by reduction of electron acceptor. Chemically, oxidation is defined as electron ejection and reduction as electron capture, as illustration by the oxidation of various ions to various e electron, Fe2 plus until Fe3 plus. Thus, oxidation is always accompanied by reduction of electron acceptor. The principle of oxidation reduction applies to a wide variety of biochemical systems and is an important concept upon understanding the nature of biological oxidation. Many biological oxidation can take place without the participation of oxygen molecule. For example, dehydrogenation is the reaction based on the law of the thermodynamic Nareswara 2013. Second, enzyme involved in biological oxidation. Enzymes involved in the oxidation and reduction process are called oxidoreductases. In following description, the oxidoreductase enzyme is defined into four groups. Nareswara 2013, namely: first, oxidase enzyme. Second, dehydrogenesis enzyme. This enzyme performs two main functions. First, transfer of hydrogen from the substrate to another in the pair by oxidation reduction of reaction. This hydrogenesis enzyme is very specific to its substrate, but often use the same coenzyme or hydrogen carrier as other dehydrogenesis enzyme, for example, NID. As a component in the respiration chain, the transport of electron from the substrate oxygen. Third, hydrogen sidase enzyme. Fourth, oxygenesis enzyme.
Number 1. The role of enzymes in the body. First, enzymes play an important role in the digestive process. Number 2. Enzymes play a role in intermediate metabolism. Number 3. Take part in accelerating reaction and breaking down large molecules into smaller molecules or vice versa. Number 4. Serves as a diagnostic tool. Number 5. Play a role in medicine. Number 6. Enzymes and medication. Number 7. Initially used in diseases of digestive disorders such as enzymes, pepsin, trypsin, amylase, or lipase. Number 8. Treatment due to lack of enzyme caused by disorders of the glands, for example, hemophilia. Number 2. Utilization of enzymes as a diagnostic tool. First, enzymes as marker of damage to a tissue or organ due to certain diseases. Examples of using enzymes as a marker of tissue damage as follows: increased activity of the renin enzyme indicates impaired blood perfusion to the renal glomerulus, so that renin will produce angiotensin II from a serum protein which functions to increase blood pressure. Second, enzyme as a diagnostic reagent. Examples of using enzyme as reagent are as follows. First, uric acid from the fungus Candida utilis and the bacterium Enterobacter globiformis can be used to measure uric acid. Last, cholesterol measurement be done with the help of the cholesterol oxidase enzyme produced by the bacteria Pseudomonas fluorescens. Number 3. Utilization of enzymes in the field of medicine. The use of enzymes in medicine includes by use of enzymes as drugs, administration of chemical compounds to manipulate the performance of an enzyme so that a certain effect can be achieved, and manipulation of protein ligand bonds and a treatment target.